Now we'll define another terminology in chemical pathology, just trying to look at all the introductory aspects of this study before we start looking at chemical pathology in depth, topic by topic. So look at another abbreviation known as the ARMS. You can call it ARMS. It's known as the Amplification Refractory Mutation System. Amplification Refractory Mutation System. Okay, so what is it and what are its applications? So it's actually highly sensitive molecular technique. So it's used to dictate specific gene mutations or single nucleotide polymorphisms. Okay. So it's particularly very important if you want to diagnose what cancer and all those genetic mutations. Don't worry, we connect it. Okay, so it relies on the fact that DNA polymerase will only extend a primer if its 3' end perfectly match the target sequence. Get the gist. So design. Two sets of primers are used, one specific for the normal and one that will usually what be used if there's a mutation. You know, see the mutation is present. When the mutant specific primer amplifies the DNA, if it is absent, the normal one will amplify it. Okay, the higher stuff. So at this level, we just want to know that something like this exists in the world. You know, you can pause to observe this. Okay, so how does it work? First, the DNA has to be extracted. So you obtain the DNA from patient sample. It could be tissue or it could be blood. DNA extraction. Are you following up? So next, the PCR setup. So use allele or specific primers in a polymerase chain reaction. Amplification. Only perfectly matched primers generate amplification. Dictation and the results are analyzed by gel electrophoresis or real-time polymerase chain reaction. So they apply it in diagnosing different disease conditions like lung cancer, colon cancer, melanoma, which will now show you the specific gene that is what mutated. Also in pharmacogenomics, they use it to now look at if a drug is not functioning well simply because there's a mutation. Do you understand? They also use to diagnose all those inherited disease condition where there's a mutation in your genes. Okay. So advantage is very specific. It will show you the exact place that there's an issue. It's very sensitive. It won't miss out. It's cost effective. It's not expensive in as much as it sounds new. And it gives you results so fast. Okay. So it can only dictate mutation. That's one of the limitations. Okay. So that's what we have for this system.